Hello, people of science. Got a really quick video I'm going to hit you guys with today. We're going to talk about RNA and DNA. DNA you should be familiar with by now. RNA, very, very similar. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. We're going to talk about how they're different, the types of RNA, and why do cells need both? Why are we learning about something that's nearly identical? So let's start just refamiliarizing ourselves with our molecule of DNA. As you can see, again, we have the sugar and phosphate backbone on the outside. With DNA, it's deoxyribose. Again, I have those nitrogen bases pointing inward, and then I have hydrogen bonds between them. Nothing new there. I'm going to bring in a molecule of RNA for comparison. I'm going to pause for a second. I would suggest you pause the video right now and just start looking for differences. All right, I would say by now you should notice that there's at least 3 differences. I would say number 1, the color is probably the first thing that jumps out at you. In this case right here, I have purple, which represents I'm looking at ribose. Instead of deoxyribose, I'm looking at a very, very, very similar sugar. Literally, it's just one atom different, but very, very similar there. But I still have the sugar phosphate on the outside. I still have nucleotide bases in the middle. I do have some new ones there, and you also notice I'm missing another strand. So similarities, here I'm focusing again, you'll notice it is still sugar, phosphate, sugar, phosphate, sugar, phosphate. That part hasn't changed. The sugar has changed, but that overall structure has not. If I'm looking at those nucleotide bases, I still have the A, the G, and the C. You're just going to notice that where I had a T before, that has now been replaced with a U. Instead of thymine, this is uracil. Overall, though, pretty similar. Here's my molecule of DNA. Here's my molecule of RNA. The 3 differences that for sure you need to know and probably be writing down. Number 1, RNA is single-stranded. There's only one side to this one here. We'll talk about why when we get to transcription. Secondly, as you saw probably right away, the color is different. I use the sugar ribose instead of deoxyribose. And finally, where normally I would have had a T, now I will replace it with a U for uracil. So make sure you know those 3 things. All right, why RNA? Well, DNA is really, really, really good at what it's supposed to do. That is storage of information. But DNA sucks at everything else. It's got those 3 important functions. It's got to store that information, carry it to the next generation, and be easily copied. But DNA is terrible at other things. Think of it as like a flash drive, something that just stores information. On its own, a flash drive is pretty useless. I mean, imagine that you didn't have any computers, but you had a whole bunch of flash drives. They would do nothing. They're just good at information storage. So you need a molecule like RNA. RNA is a less stable molecule, which is actually good in this situation, but we won't go into that. But it's a more all-purpose molecule. Think of it as like a Swiss Army knife. It's the quintessential, jack of all trades, master of none. RNA is good at lots of things. It's not great at anything compared to where DNA is exceptionally good at information storage. Like if you take a look at a Swiss Army knife, it's got scissors, but let's be honest, these scissors kind of suck. I mean, they're better than no scissors, but they're not like, ooh, I want to get my Swiss Army knife scissors. This knife here, yeah, it's good at a few things, but you're not going to be using this for, carving a turkey or tofurkey if you're a vegetarian into that. I mean, it's not a Phillips screwdriver, but let's be honest, that's a terrible Phillips screwdriver. RNA is like this. It does a whole bunch of little things okay. I mean, it's better than nothing, but it doesn't do any of them great. However, this is perfect for a temporary copy that's going to head out of the nucleus. It's more versatile, it can go more places, and it's temporary. It's going to break down. That's really, really good. So that's why we have it. All right, we have 3 types of RNA. I want you to know these. We're going to go into them in a little bit more detail. So for our purposes, we're just focusing on 3 types. We have mRNA, which is messenger RNA, tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. So make sure you have those and just a little bit of room for a blurb about them. mRNA is a disposable DNA copy that carries messages to the cytoplasm. So remember, DNA is always in the nucleus, carries messages out to the cytoplasm. tRNA, we'll do a lot more on that one later, that transfers or carries those protein pieces to the ribosome. If you remember, those protein pieces are amino acids. It's going to build a protein. tRNA brings the pieces there. and rRNA is part of the ribosome, that's the protein building factory. We're going to go a lot more into mRNA and tRNA. For now, all you need to know about rRNA is that it makes up part of the ribosome. Alright, like I said, really quick one-off there on DNA and RNA and the differences.